How many of you have ever felt ostracized because of your abnormally, almost inhumanly high intelligence? Castrated, exactly. Felt separated from the real world, isolated, filled with this desire to take revenge on the society that rejected you. I'm going to give you the means to do so. I'm going to teach you, if I can pull it up, how to create a monster. And anyone who can tell me who that is gets 10 extra points. Exactly. So, to begin with, we need some basic understanding of cell biology, because if we're going to be creating a genetically engineered monster, we're going to be using genetics. So we're going to go over some organelles, how they interact, and just the general principles of cells. Then we're going to move on to how we would isolate these key components of the cells we need to, and then how we're going to actually inject them into a new organism. And so what we're going to be doing is combining some of the unique characteristics of a little animal called Perciferous verticalis, also known as the Australian bombardier beetle. And if you're not familiar with what a bombardier beetle is, I'll explain that in just a moment. But first things first, the cell. So this is a plant. I don't know if you knew that. They're green, they tend to flower, unless they're gymnosperms. And with all plants and animals, their tissues are composed of cells. Inside the cells, there's an organelle called the nuclei. This is probably pretty redundant for you guys, but we have to go over to make sure we have a broad basis for understanding. And inside that nuclei, there are very densely condensed structures called chromosomes. Chromosomes are made of the DNA strand, which is in turn made of adenosine, guanine, thymine, and cytosine, all kind of patterned together in this long string. So, this is basically the recipe for life. Every cell has a complete set of information to create the entire organism. It's simply dependent on what parts of the cell are activated inside that DNA strand to tell you what cell it's going to be. So basically, if it's a skin cell, then large portions of this will not be being used at all, but the information is still there, which makes it nice because you can take any cell of any organism and have access to the complete and total sum total of its information. So, using that information, if it'll go forward, there we go, we take out our handy dandy little strand of bombardier beetle DNA and use these great little enzymes that are located in a lot of bacteria called restrictor enzymes. And what they'll do is come along and actually cut out the piece of DNA that you want. So in this case, we're going to take the bombardier beetle's unique ability to generate a liquid that instantly heats to about 200 degrees and then can be launched about 5 times its body length. So, by first isolating and then taking out that admittedly long strand of DNA, we can insert it into the DNA strand of a virus. And we're going to be using a virus because they have this very cool ability to enter other cells and actually manipulate their DNA. It's called a vector. So basically that's one of the 3 methods that people use to genetically modify an organism. There's also gene guns and in vitro fertilization kind of techniques, but we won't be going into that because they're far more complicated and don't have near the same success rate. So, here is the handy little vector virus that we're going to be using. You can see that there's the original DNA there and there, and then the new DNA that we introduce, in this case a gene. A gene is a full section of DNA that does one thing and one thing only. So you have a gene for dark hair, blonde hair, blue eyes, red eyes if you happen to have that strange DNA. So, as it is introduced into the new cell here, it passes through into the vesicle, which is basically the outer portion of the cell, and combines with the nucleus of the host cell. When then it starts to do its little happy dance, which involves procreating this new DNA. And so, we have our bombardier beetle, and for the sake of this experiment, we're going to be combining it with a Bengal tiger. And we're going to have the apocalypse. Your vengeance upon society. Because that's the end goal of this little experiment, of course. So you're going to wind up with a zygote, a tiger zygote, which is an underdeveloped baby tiger, basically. Whenever you have a fertilized cell, it becomes a zygote and starts to separate and divide and become a fully grown organism. And because our little tiger has the bombardier beetle DNA located within it, it's going to start using that DNA to code different proteins and enzymes and all sorts of structures. So you're going to wind up with a creature that weighs about 400 pounds, has 6-inch claws, jaws strong enough to crush a man's head, and can shoot this vitreous liquid about 20 feet. And then you have to let it sleep. It takes about 3.5 months for a tiger zygote to gestate into a fully functional adult. And after you've had your little tiger take its nap, you unleash it upon the world, and you watch it with pride as it destroys major metropoli, and just generally wreaks havoc on all those that wronged you. And since you're now apparently just a mad scientist, there's one more thing we have to get right. This is the greatest stumbling block in all of these mad scientific endeavors. The laugh. You're not giggling like a madman. It's a deep, maniacal cackle emanating deep from the chest. In fact, why not simply learn from the master? And we'll see if this works. It will not. Nope. Okay. We're going to have to do it the old-fashioned way. Ladies and gentlemen, I leave you with Mr. Neil Patrick Harris.
So that's, coming along. Strengthening the...
You need the deep. Wow. Thank you.
Awesome.